So 안녕하세요. I'm Hao-Wing, and I'm the field application scientist from GenScript's, working on the peptide portfolio. And thanks for taking time off today to come for the talk on boosting your therapeutic discoveries with peptide libraries. So for today's talk, I'll be first talking about the common bottleneck observed in many therapeutic discoveries, followed by what is a peptide library and what are some of the applications that require peptide library. The different types of peptide libraries and some of the case studies on how peptide libraries can be used in the different applications, in particular for therapeutic discoveries. I will also touch on the importance of quality peptides for us to progress with our therapeutic discoveries. So in many of our therapeutic discoveries, the identification of the target is crucial, and many of these targets are protein in nature with its own distinct secondary structure, tertiary structure, and even quaternary structure. And such a 3D structure results in the hiding of important sequences that are crucial for the function of the proteins, or for example, to elicit the downstream signaling to effect a therapeutic response. And in addition to that, due to the 3D structure, such important sequences could be spanning over multiple epitopes, making the identification and the crucial therapeutic sequences difficult, or the creation of antigenic sequences difficult. So considering such a bottleneck and limitations, peptides become a possible solutions for probing into the mechanism of action, and also act as a simple means of targeting antigenic sequences that span over several epitopes. This is because peptides are syntactically accessible and are easily amenable to chemical tailoring. They are also able to bind to shallow surfaces, which are unlike some of the chemical entities. And not only so, they can be also easily modified to non-canonical amino acid to enable changes in the peptide structure. And such changes are often enables them to be more proteolytically stable and also more cell penetrant. In addition, they are easily amenable through chemical means. That means they will be easier to increase such binding affinity through just very simple chemical modifications. So as such, to speed up her therapeutic discoveries, the identification of targets and such epitopes is paramount, and peptides could aid in this. Having a peptide library, which are presentation of multiple short linear peptide fragments, would enable such a target identification. So typically, upon identifying a bioactive region, the precise epitope target could be determined through a combination of amino acid sequences, mutations or not, depending on the issues in question. So this I'll be illustrating on in later slides on how it could be done. So peptide sequences are often presented in their appropriate assay for target identification and even function determination. So there are two major ways of creating such peptide libraries, and one is through a phage display, where the peptides is expressed on the surface of these phages upon the transfer of related DNA, related DNA into the phages. So specific clones are then selected based on the binding affinity to a target, and thereby determining crucial epitope information. One advantage of such phage peptide libraries are that they are simple and are not limited by the peptide length. However, such determination of binding affinity is often labor-intensive and are limited to natural L amino acids. However, in a synthetic peptide library, that means peptides are synthesized chemically on possibly in the solid phase or liquid phase, depending on the length of the peptides in question and also the quantity of the peptides required. This chemical synthesis allows for flexibility in the design and also the option of adding D amino acids and also require less time And in some cases, the usage of D-amino acids are desirable because it could stabilize the peptide as they are more resistant from proteases. So as briefly mentioned, peptide libraries are good with identifying target-specific epitopes. For example, if you have a protein target and would like to find out if the therapeutic antibodies bind specifically to your protein target antigen, or if you would like to optimize the binding affinity of your antibodies towards the antigen, the identification of the epitope or epitope membrane is crucial. So to do this, we can break up the antigen into small peptide sequences in a systematic manner, okay, forming peptide libraries. So the therapeutic antibodies or antibody drug leads will be allowed to bind with the peptide libraries, and the epitope could then be identified based on the binding affinity. And the same could be applied for T-cell epitope mapping. So instead of antibodies, MHC molecules could be used for this application. And it could also be used in cancer cell biomarkers. For example, a random series of peptides could be chemically synthesized, forming the peptide library. And specific protein of interest, for example, receptor protein specific for a particular cancer cell type or biomarkers from the cancer cells could then be expressed and purified and are added to the peptide libraries, and thereby identifying potential peptide drug therapies specific for the cancer treatment. So in addition, identifying such biomarker binding peptides could actually aid in gene therapy in cancer treatment. For example, like the transfer of gene, example, siRNA. For gene therapy treatment has always been a bottleneck due to the inherent anionic properties of such siRNA molecules, which render them difficult to pass through the cell membrane. So conjugating peptides with specific binding to cancer biomarkers not only target such siRNA specifically towards the cancer cells, but also aids in the cell penetration because of the cell penetrating properties of the peptides. So as such, peptide libraries are very useful in T-cell immunotherapy, vaccine development, antibody therapy, and also in cancer research. So other uses of peptide libraries are in peptide bioassays. For example, peptide libraries could be presented to embryonic stem cells to understand the mechanism of stem cell differentiation, the triggers for stem cell differentiation. It could also be used in disease diagnostics. So the assay will be similar to that of identifying cancer biomarkers, epitope identification, or it could be also used in to optimize drugs for improved antibacterial activity. So upon choosing the peptide libraries could be presented to the microbes to identify the best peptide sequence that could elicit antibacterial activity. So considering the many applications for peptide libraries, how can we generate or design a peptide library? So most importantly, we have to identify first, a bioactive region of interest to define your question. And then we can go from there and choose the appropriate library. So in GenScript, we have free online tools for different kinds of libraries to probe and answer different questions. So upon choosing the right type of a library that is suitable for your applications, all one need to do is to input your bioactive region of interest, the sequence of it, and the peptide libraries will be generated with just a click. So you can see choosing a peptide library that's appropriate for the scientific questions to be answered or the application is crucial. And in GenScript, we have 7 online library design tools, which I will be illustrating in the following slides on the type of applications each library is good for. So for an overlapping library, it is typically used for linear or continuous epitope mapping. It is a series of short peptides as shown here overlapping each other That's why the name overlapping peptide library, and the length of the peptide could be set according to how fine or the resolution one is required to define the epitope. So typically it will range between 8 to 20 amino acids, and the offset, that is the amino acid gap before the start of the next amino acid, will preferably be 2 to 4 amino acids, which is in the epitope, 2 to 4 amino acids, and the smaller the offset, the higher the resolution of the epitope. So such overlapping peptide, which aids in the epitope mapping, is good for applications such as determining the antigenic sequences for epitopes in T cells or antibodies. Such library is also good for screening substrate sequence for enzymatic proteins, or also to determine the binding site with the protein receptors. So for an overlapping peptide library, it is typically used for linear or continuous epitope mapping. And here in this slide, John and all who have published in Vaccine had actually used the generated overlapping peptide library with us for the different proteins, the PC, the GB, and also the PP65. So they were trying to develop a better mRNA vaccine that can target the CMV, the cytomegalovirus, based on the immunodominant T cell antigen. So they have earlier have developed, the targets based on the glycoprotein, GB, and also the pentameric complex, PC. But they are interested to look at, how the antigen, that is the immuno-CMV T cell antigen PP65, could improve the vaccine. And from so what they did was they generated this overlapping peptide to against the 3 different epitopes, and then they generated a peptide pools on these peptide libraries, and then they perform intracellular staining assay to probe for the IFN gamma on the T cell, and observe that yes, PP65 indeed, this vaccine could create to elicit a T cell response. But they do observe not only this test actually confirmed that this epitopenic antigenic sequence, they also determine the mechanism of action of such vaccine, in that they identified that possibly this inhibition, as noted by the addition of this a complex, actually inhibits the action of PP65, possibly through some similar antigenic sequences between PC and the PP65. So they actually use, based on this peptide library pools, they actually probe and investigated the optimized vaccine routes of application. They design a dosage kind of development in order to optimize the vaccines. So next we have, let's take a look at another type of peptide library, the Alanine scanning peptide library. So such peptide library are typically used to identify key amino acids for binding function, for protein stability, or when researchers would like to identify which amino acid is crucial for the conformation of the protein structure. So typically such input sequences require 6 to 200 amino acids, and the Alanine scanning library algorithm will systematically generate and mutate one residue at a time to an alanine, and this allows us to identify key residues in the epitopes and locate the binding site of a protein, and also evaluate the function of the protein or the enzyme. So in this next case study that we have performed in-house in GenScript's, we would like to identify key residues in the CDR or variable region of the SARS-CoV-2 antibody. And to do that, we have generated an array Alanine scanning library to replace all non-Alanine residues in the CDR region in both the heavy chain and the light chain to an alanine. And native alanine is also replaced with a serine to probe into the function of the native alanine residues in such a CDR region. So the red dots here in this figure showed the crucial residues that are important for the CDR for antibody in the CDR for antibody and antigen binding. And upon identifying such important residues, we could actually optimize the binding of the antibody through the mutations of these residues. So next we have, upon such identification of crucial residues, the positional scanning peptide library would actually allow us to do a peptide sequence optimization. So for a positional scanning library, typically upon identifying a crucial residues, we can actually systematically mutate this crucial residue to all and substitute it with all other amino acids. So this is what the positional scanning peptide library is for. And usually the input sequence will be between 10 to 200 amino acids. And this will allow us, as mentioned, to optimize the peptide binding, to enhance the antibody or T cell epitopes, and also to increase the activity of enzyme substrates. So in this following case study from Evans, Scott and al. Published in POS 1, they had actually generated a positional scanning library with GenScript at all the residues of this Cap18 sequence to obtain an enhanced Cap18 variant that is effective to different bacterial species. So Cap18 is actually antimicrobial peptides that can act as alternatives to classical antibiotics. So in this figure, the original peptide sequence is shown above here. Okay, and the respective substitution is shown here. For each amino acid. So the number here indicates the minimum inhibitory concentration, the MIC, to each mutant upon substitution with the amino acid. So for example, the red one here shown will indicate a higher MIC that is mutating this residue to the respective residue here would decrease if is effectiveness of the effectiveness of Cap18, indicating that this would be an important residues. And those with a small value of 1 or less than 1 will indicate that such residues upon or the mutation will be good because it decreased the MIC, and it would be good to optimize the therapeutic function of Cap18. As such, on the right, actually the authors had actually indicated with the following diagram, the important residues, to enhance the how they could mutate those residues to enhance the function of Cap18 as antibiotic development. So another way to optimize a peptide sequence will be through, random peptide library. So unlike, the position scanning library, peptide library, this random peptide library uses a shotgun approach, resulting in the randomness of the peptide library. So input sequence, could be between, again, 10 to 200 amino acid, and they are good for, again, peptide sequence optimization, but also for target identification and drug discovery. Okay, so in this case, from Quantararo and all published in Nature Communications, they have actually used a random library. So what they did was they fixed the serine, a phosphoserine in the middle, but flanked, 4 ferric amino acids on other side. So this gives them a big peptide library of 2 times 10 power of 8 members, allowing them to probe and identify the consensus sequence for 14-3-3 gamma binding. And as some of you may know, this 14-3-3 is a hub protein, which interacts with a range of disease-relevant proteins such as p53, RAF kinases, and estrogen receptor alpha. So these researchers in this study would like to develop modulations on such interaction through phosphorylation. And as such, because it is through the chemical synthesis of peptide libraries, the addition of non-canonical amino acid is a breeze. So this demonstrates how peptide libraries are crucial in developing therapeutic modulators. So of course, sometimes we need some kind of controls with regards to the design of peptide libraries, and scrambled peptide library could be a good way of obtaining not only the highest variation from the original library in a random manner, a large sequence permutation of the peptide. As such, it will be good when generating such scrambled peptide library to specify the library size for screening. And this, as mentioned, could act as a negative controls, but also be good for probing molecules of interest, antibodies, or DNA. So here I have illustrated an example of the scrambled peptide library that is used by Raghav and all, published in the International Journal of Medicine. So they had used the design tool in GenScript to generate 80,000 peptides with 50 peptides in one library of input length at 13. Peptide length 13. So to identify, what they did was they used this peptide library to identify synthetic epitopes, synthetic peptides that could bind to GATA1 to regulate myelopoiesis. So they would like to find out what is this and create a synthetic peptide analog that could block the interaction with between GATA1 with PU1 to allow the interaction of PU1 with C-zone to promote myelopoiesis. So as shown here from the libraries, they could identify 3 peptides, from the peptide library, and that could have a potential with a higher binding affinity with GATA1 and also but lower binding with C-zone compared to control. So from an original protein sequence, not only so, we could also generate a truncation peptide library to probe into the minimum length that is required for activity. Usually this is upon some alanine scanning library studies. We have already identified some crucial residues in the epitope, but would like to map maybe the minimal binding site for the core protein binding. So Leung and all in this published in Antimicrobial Agents and Chemotherapy illustrate how truncation libraries are created in general, and with a sequential deletion of the hexapeptides from the N-terminal or the C-terminal or from both ends of the native peptide. So this allows them to decipher the crucial sequences that are required for antimicrobial properties with a reduced toxicity. So as shown in this graph, they actually identified, using the libraries, they had identified 3 peptides, the LL, 3 epitope regions, the LL7 to 37, LL13 to 37, the LL19 that has a lower toxicity compared to the original peptide sequence, and indicated that through the decrease in the release of LDH and the improve in microbial properties of the peptides as shown by here in the PI staining here. So this truncated peptide library could also be used to identify T-cell epitope. So optimal conditions for this kind of libraries are short peptide sequences from 8 to 11 amino acids on from a MHC1 complex, or 15 to 24 amino acids on the MHC2 complex. So applications include testing all possible T-cell epitopes across a protein of interest. So to better illustrate the T-cell truncated library, Yoshi have showed here a case study from Yoshi Haiko et al. Published in the Journal of Immunology. So to demonstrate how T-cell truncated library could be used to deduce the binding epitope of the T-cell. So first, using a T-cell truncated peptide library to identify the peptide region, the epitope region, they subsequently use different peptide libraries also, such as the random library here and also the positional scanning library here, to define the number of residues, the residue that is crucial to enhance the activation of T-cell. So I have demonstrated the various peptide libraries for different therapeutic discoveries, investigation, and optimization, some of which are, used in T-cell immunotherapy, vaccine development, cancer research, and also antibody T-cell epitope mapping, and even antimicrobial drug discovery using different library design. So the table here gives, a brief summary or the possibility of the libraries for each applications. And of course, depending on the applications, the purity of the peptides required will be different. For example, for in biomarker discovery, epitope mapping, or T-cell binding assays or reaction monitoring in mass spec, typically a crude purity would be required. However, for some cell-based assay or immune monitoring, a higher purity of more than 70% of your peptides will be required. And for more stringent applications, such as you need to quantify quantitative assay, we need, at least, 80% kind of purity. And of course, in drug and vaccine development or preclinical trials or sensitive applications such as the MLR, of course, we will go for even higher purity of at least 90% to 98%. So in GenScript's, we have different options for the client to generate the peptide libraries depending on the length. We can synthesize crude peptide libraries of a crude purity. Depending on the applications that is required. The length that is required 5 to 25 amino acids. That will give a different turnaround time. So for a minimum order size of this peptide libraries will require 25 peptides, and the delivery format will be, lyophilized peptides in individual 96 well plates or individual vials, depending on the needs of the client. So this can be optimized, and of course we will give a quality check in MS or HPLC. And we allow, different kinds of modifications. We have a comprehensive modifications available for the peptides produced from GenScript. Not only so, we also have, different bioinformatic tools for generating peptide libraries. So, in additional to the peptide library tools here, like all we need to do is to input our protein sequence and then choose the appropriate library that is good for your applications, and they will just with one click it will generate the series of peptides. We also have other bioinformatics tools to allow us to understand the characteristics of the peptides that are synthesized. For example, is isoelectric points or hydrophobicity, and also the different difficulties in synthesizing a particular peptide. So in GenScript, we have, the PepH2S library platform, which allow us, to synthesize different, even big libraries for many of our clients, because we have we can synthesize peptides in a high throughput manner at 18,000 peptides per month, because we can guarantee fast turnaround time, because this is run 24 hours, and it is fully automated, allowing a reliable quantity and quality. Not only so, we have, the AccuPep QC, which has, different kinds of QC that is available for the client. For example, endotoxin control, or TFA counter ion removal, solubility test, peptide sequencing, and also LC-MS analysis. And we are certified for ISO 9001, such that our quality of the peptides is certified. So not only so, we understand many of our peptide libraries are also being used in, cancer research and also T-cell immunotherapy. That's why we have collaborated with Cayman to provide, immunopeptidome profiling, which is a deep sequence analysis of the MHC-associated peptides to allow clients to identify new antigens and potential immunogenic sequences. So what we typically do is we lyse, the cells or tissues collected from tumors, and then we perform a NHC immunoprecipitation to collect these peptides that are bound to this MHC class 1 and class 2 molecules. Upon that, we will run an LC-MS to identify the MHC peptides. And after that, we can generate such new antigen peptides or peptide libraries, for client to work on their cellular assay. So we also understand, some of the difficulties in synthesizing such new antigen peptides, because sometimes such length, they will span between 8 to 50 amino acids, and sometimes peptides over 21 amino acids are difficult to synthesize, and also those neutral peptides are also more difficult to synthesize, especially sometimes such peptide can be hydrophobic, which increase the difficulty in synthesis, and also its purification due to its inability to solubilize in water. And so at times such clients need a high yield of these new peptides for the assay. So we also need to ensure the yield and the purity of these new antigen peptides. But in GenScript, we have, the technologies such as the NeoPre algorithm for us, to actually input the sequence and actually identify, the difficulties in synthesizing such peptides. And then from there, we can advise on, the type of synthesis technology that could be used for such difficult peptides. So because of that, we have increased in, the success of synthesizing peptides. And we also have the Hysing process, which is optimized reaction. In which we have optimized the reaction temperature and also the condensation reagents, and also increase the coupling efficiency, such that we can get using this Hysing technology, we are able to increase our success rate to 91% to synthesize a difficult peptide, such as those hydrophobic peptides of very good purity. So we also have a double coupling technology, which we actually use. We have optimized the using of coupling reagents from Hysing technology to be used with optimized reaction conditions in the microwave technology. And we are actually the first CRO with this double coupling technology, allowing us to increase of the success rate of those initially difficult peptides that are unable to be synthesized using normal means. Okay. So with that, as an ending note, I hope I have given you, a brief summary of, what peptide libraries can aid and in our therapeutic discovery, and how GenScript will be able to give you, quality peptides, to synthesize for your peptide libraries for your research. Yep. With this, I thank you for your time and attention.